presentation today is Zachary Graff. He's from Southeastern University, and he's part of the Stony Coral Tissue Loss Disease Resistance Research Consortium Regional Project. And he's going to be talking about improving Orbicella faveoalata restoration efforts by examining tissue regeneration and fish predation after coring.
Hello everyone, my name is Zachary Graff and I'm a graduate research assistant at Southeastern University up in Dania Beach. I'm going to be talking about a little bit of research the lab has been doing over the past few years. The title of this presentation is Improving Orbicella faveoalata Restoration Efforts by Examining Tissue Regeneration and Fish Predation After Coring. I cannot read and lean at the same time. As we all know, coral reefs are pretty important. They provide some of the most biodiverse ecosystems on the planet and are a habitat for around 25% of marine life, while also providing vital services to our state, including storm surge and erosion protection, as well as generating a great deal of income via ecotourism, recreational, and commercial fishing. Yet unfortunately, there has been a drastic decline in coral cover over the past few decades. This decline is largely due to local and global stressors such as a reduction in water quality, increased public usage, and the effects of global warming. These stressors contribute to and increase the severity and frequency of bleaching and disease events. Curiously, not all corals are affected by disease equally, and little is known about the varying degree of susceptibility amongst individuals. In recent decades, declines in coral reefs globally has increased the need for more research to be conducted to understand the cause of coral reef declines, to mitigate for those causes, and to inform reef restoration. These are a few examples of increasing publications through time from the Arabian Gulf and coral reef modeling publications. Simultaneously, a new trend in coral reef research has emerged with the use of many new molecular techniques now available to study functions in coral tissues. Although incredibly useful and necessary, these new techniques impact coral by removing live tissue and skeleton to capture the holobiont tissues. The combination of increasing coral research and potentially more destructive sampling methods warrant some attention. Coral disease has been associated with tissue wounds, colony fragments, and predation scars. AB and Santavi found that injuries significantly increased disease susceptibility in black band. Brandt et al. Found significant increases in disease on fragments versus attached corals after a bleaching event. Many studies have found associations with feeding scars and disease. Although the mode of transfer is unclear, it is most likely from the predator transmitting the disease directly or the coral becoming infected indirectly by predation wounds. Coral diseases are drastically shifting populations at local, regional, and higher scales. Studies have shown that rising sea temperatures are likely to increase the frequency of disease outbreaks affecting reef-building corals through impacts on coral hosts and pathogens. Therefore, it is important that we investigate any associations between recent coral sampling and disease. Investigating coral sites can help provide valuable information on coral health. Tissue regeneration and wound healing is an important indicator of coral health. Healthier corals would have more resources to devote to regeneration and hence grow faster than stressed corals. Coring corals can also leave lasting damage, so understanding those outcomes is important to help mitigate sampling impacts. The goal of this study is to investigate tissue regeneration and disease associated with coral core sampling to understand individual coral health and how different factors may be impacting a coral's individual resistance to stony coral tissue loss disease. Our lab is collaborating with other researchers to investigate SCTLD resistance and susceptibility in Orbicella. This group is called the SCTLD Resistance Research Consortium, or RRC for short. The goal is to understand why nearby corals have varying levels of resistance to disease. Catch Brian Walker's talk for more details at 4:45 today. Over 1,700 cores were collected over 3 time points at the same location on corals and sent to our expert labs to analyze the cores to gather a snapshot of as much information about the holobiont as possible. The hope is to identify functional relationships between genomics, transcriptomics, metabolomics, and proteomics and the holobiont to identify differences between these resistance groups. Here are some examples of that massive undertaking. Our team was very efficient and ensured that all samples were collected and preserved within 15 minutes of removal from each colony. 90 Orbicella colonies were sampled using a stainless steel leather punch, and we removed multiple small quarter-sized samples over 3 time periods, 45 in Southeast Florida and 45 in the Lower Keys. 8 cores were collected in May and June, 5 in August and September, and 6 in February and March. Core sites were monitored every 2 months using a standardized framer. ImageJ software with a lesion macro from NOAA was then used to assess area and perimeter of these lesions. The program measured area and perimeter of each hole of the parent colony and summed all of these areas together from the same colony for the survey and monitoring periods, and then calculated a daily rate of change in each area. This quantified the amount of non-living tissue at the core sites. Data was excluded from the analysis if a type 2 lesion occurred, meaning that a lesion merged with another area of the core that lacked tissue. This occurred in 5 of the parent colonies. If disease was present, the coral was removed from the area analysis to prevent any confounding variables. This occurred in 4 of the parent colonies. This study evaluated the first 2 sampling periods and the first 3 monitoring periods. In the first sampling period, 8 cores were taken, then the holes were left unfilled. In the second sampling period, 5 cores were taken, all holes were filled with modeling clay, including sample period 1 cores. Tissue area differences between time points were standardized to a daily change to account for differences in time between visits. The factors: colony location, region, and size, filled versus unfilled, and core site angle and location on colony. Only 4% of the core sites had disease. It is yet to be determined if the disease arose at the core site or started in another location on the coral and then came through the site. Continued monitoring will give us a better understanding of disease associated with core sites. Note the y-axis. Note the y-axis units are daily change in non-living tissue in millimeters square per day. So higher values mean less live coral. Florida Keys corals had a greater daily change than the ECA, meaning that non-living tissue expanded more in the Keys corals. Subregion analysis showed high variance in South ECA. You can see the green up here, and that is Miami and Key Biscayne, and tissue loss was greatest in the purple and Sand Key. Unfilled 1, green, shows the impact after the first monitoring period, while unfilled 2, blue, shows the impact after the second monitoring period, meaning significant tissue growth between monitoring periods for the unfilled. Contrary to the unfilled, the filled core sites, red, exhibited significantly lower areas of non-living tissue, meaning that they were healing after the first monitoring period. As far as the angle on each coral that these cores were taken from, we designated flat as 0 to 20 degrees, that's our red. An angle was 21 to 69 degrees, that's our green. And vertical was 70 to 90 degrees, and that's our blue. Core sites with all angles had increases in non-living tissue. However, vertical sites and sites on the edge of the colonies had the highest tissue loss. Colony size did not play a large role, even though it was hypothesized originally that small corals would have a higher predation due to the lack of available tissue on these small corals. We do not see significant differences between hole position or hole locations. So what happened? Why so much tissue loss at the core sites after coring? Well, it appears that our coring facilitated heavy parrotfish predation at the core sites. Revisits showed extensive areas of the core edges were chewed away. While we did not physically witness this predation, Visual cues at the sites were consistent with parrotfish predation. At this time, we do not know for certain which species contributed to the impacts. Reef fish play a critical role in maintaining a healthy reef ecosystem, yet heavy corallivery can often negatively impact already stressed coral colonies. Corallivery can create colony damage, forcing corals to deplete energy reserves on repair rather than growth or disease resistance. They can also act as a vector for disease from one coral to another. It has been particularly damaging in South Florida coral restoration efforts, where parrotfish eat out plants and negate months of restoration efforts. After noticing these impacts, we decided to fill all of these sample holes with modeling clay during the next period to try to mitigate for these impacts. Filled cores had significantly less predation. There could be a variety of reasons why unfilled cores were predated on at a higher rate. Scent, bacteria, color. It has been suggested that corallivorous fish may be attracted to the endolithic algae in coral skeletons or the scent. Other studies have found increased predation to exposed skeleton and fragments. Exposed edges could also make it easier for predators to find purchase and to bite into these colonies. Future studies could explore filling in the holes with a variety of different levels to test whether the bare skeleton versus endolithic algae makes a difference. We also saw significant differences between the two unfilled groups, suggesting that predation decreased over time. We hypothesized that this may be due to a reduction in chemical and visual cues due to healing or the holes being covered by skeleton or algae or sediment. This was an interesting result as it followed trends from other outplant studies in which researchers observed a reduction in predation over time. It will be interesting to see in the future how the two groups of clay versus non-clay filled core samples compare in terms of regeneration over time. Regional differences in tissue loss indicate a difference in predation between the Lower Keys and Southeast Florida. In particular, at Sand Key, different fish populations in the two areas might explain these differences. Curiously, flat surfaces had the lowest daily change, perhaps giving additional cues to how to avoid future predation impacts. This follows trends found in coral outplant studies where researchers used the crown method to limit fish predation. From this study, we recommend that similar tissue sampling projects remove core samples from flat angles if possible and immediately fill the holes upon core removal. We are currently analyzing other monitoring periods and the sampling impacts after the third sampling timeframe. Future research will evaluate tissue regeneration for temporal differences, regional differences, and association with the SCTLD-RRC project data. Quick thank you to all of our partners. Thank you for being here today, guys.